Hello everyone, and welcome to this AsiaEdit Author Success webinar. I'm Trevor Lane, an education consultant at AsiaEdit. For more than 25 years now, I've been helping researchers to publish their scholarly work in international journals, and I've taught research writing to thousands of researchers worldwide. In this webinar, we look at how best to present data in manuscripts as tables and figures. Now, they're not just illustrations or decorations, but a key part of research communication. So in this hour, we'll cover table preparation, figure preparation, and my top tips for presenting data in manuscripts. The final 10 to 15 minutes of this hour will be reserved for a Q&A, question and answer session when I'll answer any questions you may have. First, a word about the Zoom functions. To make the webinar interactive, I'll be asking you to use the raise virtual hand button and to type in the chat box. But if you want to ask a question for the Q&A session, please use the Q&A question box and not the chat box. And you can type questions in the Q&A box at any time or during the Q&A session itself. So let's start with tables. Now, I have a quick quiz for you, and I want you to please type in the chat box when you're ready. So out of these display items, which do you think is better? So A, B, or C? So please type in the chat box which you prefer, what conveys the best information, A, B, or C? So someone's put A, a few of you have put A. It's scrolling up quite quickly. A, B, oh, now C is going up. So it was A to begin with, a few C's. So I think A and C is about 50% each. No one chose B. So let's have a look. I think I agree with you for C maybe, but let's have a look. So let's look at the names of these types. So it's the same information, but different formats. So A is called a pictogram or an isotype. So we see it's like a graph, but it's actually got car images, car icons as the bars. B is a 3D bar chart. So bar charts we know, but the top of it looks like it's got a 3D layer. And then C is a table. Now, A is okay, but maybe just for magazines and newspapers and websites. So things that the general public will see. But for, of course, an international journal, it's not quite right. It's a bit too simple. So cartoons and icons for that type of graph is not suitable for journals. B, bar chart is okay, but a 3D bar chart, you can see at the top where the person's added kind of 3D perspective. We actually don't know from the axes, is the bar pointing at 2 and 2.5 and 3 and 3.5? So that's a problem with 3D bar charts. They cause more problems than they solve. So stick to 2D. And then the table is normally okay, but it seems a waste of space for just two numbers or a 2x2 table. So actually the answer here, I would say none of them are better. It would just be best to use text. The sample consisted of 2 blue cars and 3 red cars. That would be clear enough. So sometimes text is better. So do consider what's the best format, figures, tables, or maybe just text. But don't repeat the information in many formats. So here we have some simple tables, and we normally would expect tables to have numbers in, but actually this is a type of table too. It's a text box and it's just a gray background, sometimes light blue, so a light tint. So I'd use maybe 10 to 15% tint of a color. Don't do 50%, you can't read the text then. And we'll notice that it's an extract or quote in this case. So if you're quoting from a person in an interview and it's quite a long speech, or you're reproducing a paragraph from a report, of course you have copyright permission to do that, then it would go in a box. And you notice that the box number and the title is inside the box, not outside the box. And it would depend on the journal style. If there's only one box, sometimes they just say box, sometimes they call it box 1, even if there's only one box. And below is the active data field. So it will be in a tinted box, and it will be text big enough to see when it's shrunk down, and decide if it looks pretty as left justified or left and right justified. So here it's left justified, right ragged margin. It's not completely square, the text. It's not flush both ends. So it sometimes looks better this way. If you force it to go along the whole line, you end up getting funny gaps in the text. So watch out for that. See what the journal prefers. Now, there's also tables with just one list of a column of words. So here we have a list table, and they're useful for criteria, summary points, categories and examples, or themes and subthemes. And we see the table number and title is similar to the one in the box, but it's at the top. So it's outside the table, and it says table 2 and then the title. Again, if there's only one table, you check with the journal guidelines if they just call it table or table 1. And why is it at the top? Because for tables, we usually read in English left to right and then top to bottom. So people will notice the title at the top of the table. Now, in this case, we have rows with rules. So those are lines going across. And there's shading as well, separating the main titles. So again, check with your journal. Sometimes they don't like rules going across every line. Maybe they do it every 5th line, or they have shading, a lighter shade on alternate lines. And again, check the shading isn't too dark. And here we notice there's 2 levels. It's a hierarchical table. There's 1 and then 1.1, 2 and then 2.1. So it's a 2-level nested list because it's embedded. One list is embedded in another list, and there's indentation to help readers see that there's a sub-list. In this case, maybe the author could have formatted the column of text as just one column, as 2 columns, and split it up as the 1, 2, 3 level, and then the second column as the 1.1, 2.1. So again, see what works best. But maybe for space, there wasn't space in the journal for 2 columns, so they squashed it into 1 column. And it could be a bulleted list as well. Here it's a numbered list. And it's useful then for article highlights. Sometimes a journal asks for the 5 most important findings. So even that would be in this type of format. The groupings are in a logical order, so don't just do alphabetical order. So think of how the reader would find your table the most useful, maybe group by most important to least important, or different directions, northeast, southwest, southwest. So think of a logical way to group and order your text. And then we'll notice at the bottom, 6.1234, the grammar is parallel. So keep the items in each of the list as similar grammar. So we see these are all nouns: awareness, accountability, not for, I can't read that, and the value. So these are all nouns. Don't swap some to be verbs or ing or adjectives. Now, next we do have another form of text table. It's split up into more columns. It's more complicated. So this time the column heading is at the top there, source of genetic variance, but we see it's a nested column. So one column is split into 2. There's the cohort and then participants. So you might have to do that for one overriding column heading and then subcolumns. But again, use a logical hierarchy and make it nice and visible and beautiful, and align everything well. So here we can see that sometimes tables have references. So here there's the Vancouver numbering style. So in this case, the references are important, but you'd use the same order of numbering as what happened in the text. So where the table is in the text, the number before would have been 1 before this one, and then you add the number here. To be safe in the text, when you say see table 1, you'd add that reference as part of the sentence so that the numbering doesn't go out of order. But sometimes we'll see in the next example, there's many references which may or may not appear in the actual text. So you'll see a jump in the number. So that's okay, but use the same ordering as the text. And then we see there's a row heading on the left. So the first column is actually the names of the rows. And we see there's also nested rows. There's sub rows inside the rows. So again, organize and embed as you see fit. Now, an important part of tables is the footnotes. And we'll see at the bottom, it's below the actual table frame. And in this case, it's actually the same size as the text in the table. So don't make it very small, because it will get shrunk again in the print journal. And we'll notice what the footnotes are for. It's for things that don't fit inside the table and can't go in the heading. And we see it's a data source. So sometimes you'd use a source of data from the government or a website. So give the source, give the link, and maybe say you have permission if you need permission. Sometimes a table would be reproduced or adapted from a published one. So you might have to do that, but you need copyright permission. And then you have a little credit line saying you have permission and thank you to the people that gave permission, and you give the reference. Or here, there's also a link for supplementary information. Nowadays, more and more journals use supplementary files because the table is only a section of more data which is available in the supplementary files. And most importantly, you explain your abbreviations. So just put the abbreviation, then the full form. Sometimes if it's a small list, it will be listed one after another. Here it's one after another in a continuous sentence. So it's more difficult to see, but your eyes will get used to it, that you look for the capital letters and then you see the definition. So we see that different types of footnote have their own line. So the source was in a separate section and then the abbreviations in a separate section. Now, another form of simple table is not quite words, not quite numbers, but symbols. So you might have a grading system or a scoring system where you have plus, 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 or here we have different traffic light symbols for risk of bias. So that's okay. So this is sometimes called a matrix, but it still is a type of table. And we'll notice that the column headers, in this case, they're called the headers, and they're independent variables. So the ones that the researcher is interested in. So they didn't measure it, but they controlled these ones themselves. Similarly, the left-hand side is the row headings, which is called the stub, or here it's the groups. And again, we use, in this case, they've used a systematic review. So they've put references, and they've used the Harvard method, name plus year. So actually here, the numbers won't jump in the main text because the author names and year would just appear in the reference lists. So it doesn't look strange in the text. Now, these symbols would need to have a key, sometimes confusingly also called legend. So here the scoring system is explained. And here the scoring system is the risk of bias, which is the dependent variable, the one that varies or the one that is measured because the independent variable changes. So sometimes this variable is listed in the header, in the column heads, but sometimes it's only in the title. So be careful also when you read a paper, you know what the dependent variable is in the columns. It's sometimes hidden in the title. And as I've said, there's in-text, in-table citation, and then grid lines here. So see what the journal prefers. They might say it in the author guidelines, or look for some past samples to see what the authors have done, what's acceptable. Sometimes it's easier to see if there's grid lines going across and down, but the width has to be fine. So it's not like a 1 or 2 point line. It's 0.5 or 0.25, we call that a hairline. Or sometimes journals don't like grid lines going across and down. Maybe they just want the ones going across. So watch out for that. This table is mostly text, but there's one column of numbers. So this is a mixture, and also mixed data would use this format. So here we're presenting themes and sub-themes and some sample quotes. And the table legend is at the top, which is sometimes called the caption, which is the title plus important information, but actually try to include the extra things in the footnotes rather than having a top-heavy title. The column here is quantitative data. Everything else is qualitative because it's text, and the author has left aligned all the text to make it look nice. And we notice there's a few rows per main row, so it's nested rows. But the title, I'd say, is a bit strange because it says sub-themes and then it numbers 1, 2, 3, 4. But we see that the 1, 2, 3, 4 hierarchy is theme, not sub-theme. So the author's got it wrong there. So be careful when you describe what's in your table. Sometimes you spell it out like here. So I've put a better title, themes and sub-themes, comma, with sub-theme frequency and sample quotes, and then say where it comes from or the people it comes from post-training interviews on patient experience. So that's a better title. You don't have to list every row, every column. If there's a few countries, you just say by country. You don't have to say according to India, China, UK. Next example is numbers. So we call this a data array or data table, and there are data values, multiple categories. So this is the usefulness of tables when you compare multiple categories at the same time, going down and across. Now, tables make you think about the type of data. So of course, hopefully you know the types of data from the analyses. So categorical variables are groups here in the rows, and each category is split into smaller ones. So these are called bins, data bins, like throwing things in a bin. But of course, you have to think of what bins you use quite carefully. And this case for age, even though it's a continuous variable, the ages have been split into categories. And because it can go upwards, it can be ordered, it's called ordered categories. And or ordinal interval data, because we've made intervals out of continuous data. Now, for continuous data like height and weight and age, it's best to summarize the distribution as a central value, so mean or median. So here we have the average or mean as the central value, and then the spread or the variance, which could be SD here for standard deviation, or standard error of the mean, or 95% confidence interval. So be clear which one you use. And then this is an example of binary or dichotomous variable. There's only two choices. So yes and no type of categories is binary. And here we have an example of 3 or 4 categories, but you can't really order them. So they're non-ordered categories. So this will be something like hair color, black, brown, or blonde. You can't really order them. And here the numbers are clearly in the same format in each column. So the numbers, the whole numbers in the N are called discrete data because the whole counts. And then in the percentage is not discrete. But here the author could have maybe collapsed as one column N and then brackets percentage. So sometimes you see that as one column, but because they have the space, they've put it into 2 columns, and they've centered the numbers. And sometimes journals want you to center around the decimal point for easier reading. So beware of that. And if there's percentages or N's, don't forget the whole number, the denominator. So capital N here is in the title, 104. So that's a common mistake that authors forget the complete whole number N. Another table of numbers is called a crosstab or cross tabulation. Now the numbers are small, it's not really for reading, but it's for multiple comparisons left and right. So we have nested tables, nested rows, nested columns, and it's useful for multiple categories. Large tables, sometimes you might have to turn the page, if it's a print journal, into landscape, and sometimes you might have to split the table into two and go over the page. So hopefully the journal art department will help you and then split it and put cont for continued at the bottom of the first half, and then table number cont as the heading of the next page. So the dependent variables is the one that you measure. It usually goes on the column head. Don't forget the scale or units. If you have something like millions, you don't have to put all the numbers in the column. You can just put times 10 to the 6. So here we have per thousand, which applies to the whole column, or percent, which applies to the whole column. We have independent variables embedded as columns. So this is the nested columns. So independent doesn't stay on just the first column left-hand side. This is why these crosstabs are useful, because you can split subgroups of independent variables also as columns. So here we have as race ethnicity. Now, the advantage of tables is that you can put the overall characteristics for the whole group or for the whole population, or you can total columns or total rows, or find differences and put a whole column as a difference. So that's easier to see. That would be very difficult to put in a bar chart. So in this case, most of the independent variables go in the first column as the row headers, and it's nested rows, and the author and journal have chosen to left align all the numbers in this case, and its central values as mean and 95% confidence interval in brackets. Now you see NA, which could mean not available or not applicable, so do explain that in the footnotes. So here NA is not applicable because the testing date isn't important. But do make sure you put the right type of abbreviation. Why is the data missing? And also, if data is missing but you used an imputation method to estimate missing values, then you have to explain that too. Statistics, apart from descriptive central values and spread, they can be inferential too. So here we have p-value that compares columns of data for statistical associations or differences between groups. And here we see it's the risk of severe COVID-19. So it's a case control study. It's a retrospective one. We have columns for cases and controls. And in this case, there's continuous variables, ones which go on the sliding scale. That's the independent variable with units, and then give the summary statistic, mean and SD. And there's also categorical variables, and again, give the units as part of the header in the row. In this case, the data are right aligned, not left, not centered, and the confidence intervals don't have a dash or a hyphen, or sometimes an n dash, the width of an n. So it's a wider hyphen, but sometimes you have negative values. So some journals prefer 2 TO rather than a range. And in this case, the dash or the n dash doesn't mean missing data. It's convention that for odds ratios, you need a reference category. Sometimes authors put REF, ref, but again, be clear what you're doing. And here it's raw odds ratios, so be clear if it's raw or adjusted. Finally, I want to talk about footnote symbols, but here I want to point out that the authors used consistent decimal points, decimal places. So even 44, even though 44 is a whole number, they've put 44.0 to make sure that everything has a point something for one decimal point, one decimal place. And I said an advantage of tables is you could do differences, so they could have added a difference column here. But the p-value was to compare the columns. But the footnote here, we notice that the important footnotes go first, again, before the abbreviations, and the authors used little letters ABC, and the order of footnotes goes, remember, it would go across a row before you go downwards. So the order of footnotes is going across the row, each row down, and then successively downwards. You can use capital letters, little letters, numbers, Roman numerals, and they link to things like conditions and variables and statistical tests and results, and sometimes abbreviations, but it gets messy if you use a footnote symbol and an abbreviation. So usually the abbreviation will do, but there are some special footnotes you might come across. So they're symbols, and it's always in a special order of asterisk, then dagger, double dagger, section mark that looks like two S's, paragraph symbol looks like a backwards P, parallel mark, and the number sign. And then if you go beyond 7 footnotes, you start doubling up and have 2 stars, 2 daggers, 2 double daggers. But of course, you shouldn't be using more than 14 footnotes anyway. There's probability symbols for 1 star, meaning P is less than 0.05, 2 stars, P is less than 0.01, and 3 stars, P is less than 0.001, and then that's usually the minimum that you go to. So that's tables. Let's look at figures. So again, please type in the chat box, which is better, A, B, or C as the figures. So I see some Bs. So this is going to be the dates, milestones of COVID-19 pandemic for Germany. So some Bs, C. So the dates, but I mean, we wouldn't really show them as a calendar, but actually none of them are suitable. Maybe B would work, but it's difficult to see. So something that is on a timeline can use a timeline, and then you can show periods as well. And sometimes for schedules and timetables, we can use something called a Gantt chart, which shows different activities, and then the bars are the timings. So there's many types of figure, of course, and some figures can be chemical formulae with pathways like this one. So it shows special arrangements or sequences. Now the figure or legend this time is at the bottom. So put your hand up if you think that this is okay, or type in the chat box if you think the author's made a mistake by putting the title at the bottom. Yes, actually, it's okay. So the convention for figures is to put the title at the bottom, and we'll see why in a minute. So tables, the title goes at the top, figures, the title goes at the bottom. And also explain in the title, it doubles up as the footnote. So explain any symbols. So here you can just about see there's a red star, so that gets explained there. There's other types of figures for molecules. So there's ribbon diagrams, and here you can have more than one part of a figure. So those are called panels, and then you label them A, B, C, D, E, and explain it in the figure legend. So it's quite big, the figure legend here. And even circuit diagrams would be classed as this type of figure. Now, flowcharts you might use to show the flow of in a method, or flow of patients, or a decision tree diagram or algorithm. So it shows functions and relations, and the processes and instructions, decision trees. Organization charts are usually boxes and lines or arrows. So these are box and line diagrams. And the convention, again, for decision trees is to have a diamond as the decision points. So go online, work out the logistics and the conventions of how you use the symbols and boxes. Here's another example of a specialized one, a path diagram. So it shows cause and effect, or impact, or network analysis, or relationships. And the convention is to have the variables as the rectangles, error terms as circles, and then the directions and the size of the relationship is in numbers and arrows, joining up the shapes. You might see a spider or radar or star chart. So this is multivariable diagrams, more than one variable, multiple features for a single observation to track something like performance and commonly qualitative data. And the spoke or radius is where the variable goes. You might see in qualitative studies an overall kind of mind map where the theory is shown in arrows and shapes. So it's a schematic diagram. And it could be the theoretical framework either underlying a study, or it could be the theoretical model that the authors create from their study in the hypothesis generating paper. Now, this is more familiar to many of you. It's a dot plot, but it's simplified data of only the central value. And so there's two symbols, two data to each category or each variable. So here we explain it in a symbol key, and we label the axes clearly, of course. And the main dot is the mean, and it has error bars either side with a little tick called a cap. And here the independent variable normally goes on the x-axis, the horizontal one, but it's you can change it. So here it may be a questionnaire item, a questionnaire sentence is quite long, so they've changed the axes around. So as long as you're clear and you label the axes, it doesn't really matter whether it's on the X or Y. And the legend is at the bottom, as I said, and the authors explain the statistics, but it's a block legend because we don't use separate lines or sections. It's continuous text. So it's quite difficult to read, but again, that's the convention. And in this case, we compare the two values in the pairs, where the p, there's one asterisk, so that must be p less than 0.05, and you use a bracket to show which shapes you're comparing. So for continuous data or trend data, it's best to use line graphs and scatter plots. So you join up the lines to show trends and relationships between the X and Y axis. And again, don't use solid grids like in Excel. Some journals let you use hairlines for horizontal grids, or very fine hairlines if you use the complete grid. So in this case, the scatter plot is actually all the data plotted, and then you join them up to show whether they're correlated. And here the grid is actually both directions, and the legend will explain the data key. Now, the bar chart we just saw usually is for, or the dot plot we saw is for normal data. So it follows a bell curve, a Gaussian curve. For non-normal data or non-parametric data, you have to use a box and whisker plot. And this also has a special meaning. So the boxes, the little colored boxes, the height is the interquartile range, which is the middle 50%. So it ranges from the 25% to 75% mark after you've ranked your data. The line in the middle is the median, so it's the central value when you've ranked it, so the 50% mark. And the error bars are shown either side, but again, it varies. Sometimes it's the complete range from lowest to highest value. But here we see some outliers where the dots go beyond the error bar. So sometimes it's 5% to 95% range, or sometimes it's 1.5 times whatever the interquartile range was. So sometimes it's not the actual minimum maximum. So explain it in the footnote. Here they've used 5% to 95%. Similar to box plots are violin plots, where there's a width involved. It's the probability density. So it shows the amplitude and distribution of the data as well. Now, bar charts are useful for more than two groups. So before we saw the dot plot, which had the mean and SD, if it was just a pair of data for each category, then it's easy to see and compare them. But here we have three categories. It would be difficult to do that here. So we use bars, even though one of the bars is 0. So it correlates to a gel picture. It could be Western blot or some kind of electrophoresis gel picture. Now, when you show gel pictures, you can't stick columns together and then try to smooth out the joins. You have to show where the joins are, and then you have to be prepared to show the original data to the journal because it's too easy to cheat. So for gels, be very careful. And for bar charts, it's useful for multi-series. You quickly compare more than one category in parallel. And again, this is what I mean by a block legend. Everything is in just one block, even though it contains things like methods, results, error bars, how many replicates and p-values and abbreviations. It all goes just into one block. Check the journal. Sometimes you do have to include some results and methods details because they want short method sections in the text and then lots of methods details in the footnotes, in the legends. Here also for continuous data, we have histograms, which is a special type of bar chart, and it shows frequency distributions. And again, you put the data in intervals. And here we have left and right mirror images. So sometimes it's called a population pyramid for male and female. And the one below is called a stacked bar chart. So it shows the distributions as the percentages of 100%, but because we don't want to use pie charts. So pie charts are banned in many journals because it looks too amateurish and it's difficult to compare pie segments, the different angles. So it's better to use stacked bar charts. And in this case, there's a two-dimensional version. There's an extra meaning to the width. So this is called a mosaic plot or a Meko chart. And again, the legend gives the complete total N and the data source here. Now, if you want to use color, you can have a cluster heat map. So here's the correlation matrix for variables which are repeated on the X and Y axis. So the diagonal would be where it's +1 positive correlation because it's actually the same variable on the X and Y, and explain the color coding in a key. Similar to color maps, heat maps, is a choropleth map. So you might have seen this for COVID frequencies. So it's the variable that is corrected for or calculated for population density as well as region. And again, explain the key with an interval. Finally, we come to tips for presenting the data. So I'm going to talk about a few more types of figure and also how you describe your data in text. So a quick quiz for you. Here's equations. So please type in the chat box, what's the best format for equations? Is it like a table text box, or you call it a figure, or put it in the shaded box, or draw a square around it? How would you present formulae and equations? Well, you wouldn't really put it as a shaded box because there might be many equations. So actually, this was a trick question. Equations normally go as continuous text, but you just put it in a new paragraph. So this is not a figure. You sometimes number them and you put definitions. Sometimes it's continuous in a sentence. Sometimes it's split up here and aligned for easier reading. If it's a short equation, it can go right inside the sentence even. So this is also similar to social sciences studies where you present a quotation. If it's just one sentence, you put it in quotation marks inside a sentence for what someone said in an interview. But if there's more than one line, you set it off as a separate paragraph, maybe smaller font in its new line, sometimes as a block quotation. But then for bigger quotations, you do use a box for that, or you might even use a table, as we saw. But so for quotations, you can use a table, but for equations, usually not as a table. Another type of special text which you don't present as a table or figure is chemical characteristics, a melting point, boiling point, NMR, peaks. Again, that goes inside the text, but just as its own paragraph. And sometimes you might want to present text as a figure. So there are word clouds. That's a pretty way sometimes to show frequencies and importance of terms, especially in social media research. Now, you do have human photos too. So here are X-rays. For actual pictures of people, you do need, and even for X-rays, it's best to get patient participant consent. So participants need to give consent for participating in the research, as well as giving permission to publish the research, because they might get identified from some of the details. And they also need to give permission if you want to share the data, even though the data are anonymized. So many journals now want you to share raw data. So even real figures, real photos can have features marked. So there's an arrow here, and then you explain what the arrow means or is pointing to in the legend. And then other types of figures could be brain scans and machine traces, so actual tracings from machines, and annotated drawings like plans of a room, and it could get magnified. So here's a circle and then a magnified portion, or we call it an exploded view, where a contraption, a device is shown in its parts, but kind of exploded out so you can see where everything fits, kind of like an IKEA picture. Now, another special type of social sciences presentation is discourse analysis or conversation analysis. You'll see that every line, every word has been typed out with numbers for the seconds of pauses and symbols for when the two people are overlapping in their speech. And sometimes you show when they're whispering, when they're shouting. So this is special. Sometimes you show a sample inside the main text, but then you put the rest of it in supplementary data. So this is about supplementary files, and see what the arrangements are for the journal and also what they call it. Sometimes it's called appendix, sometimes it's called supplementary appendix or appendix supplementary data. Sometimes you just put the link of where the data can be found. So you've uploaded it to the journal site or to an external public repository. And sometimes you say whether the data are available. So if you've used a public sequence data, for example, DNA or protein sequences for the National Library, for the NCBI, then you have to give the link and the actual code of how you uploaded your data. So you do have to share your data these days. Now, a little word about the artistic side. So if you have multiple panels, do lay out your figures in a nice, pretty way with lots of white space, so it's comfortable for the eyes. It could be balanced, it could be unbalanced, it could be symmetrical or asymmetrical. You could imply a direction to show where the viewer should be reading down the page. It could be small and large, different scales. You could try to make it regular grid pattern like this, or make it different on purpose. Sometimes you can't control it because you have a small picture, big picture, and if you magnified everything, it wouldn't look right. So just take an artist's eye when you do this, and be careful of the color you use, because sometimes the colors can clash and make it very difficult and uncomfortable. So colors that are opposite each other in the color wheel, color spectrum, are called complementary. So red and green, orange, blue, purple, yellow actually hurt the eyes when you put them close together. We'll see in a minute what that is. Colors that are close to each other, so similar oranges, similar reds, those are called analogous. So like a scale of colors. As long as the colors are far apart from each other, you can use these in bar charts, as long as they look different enough. Now, colors that are opposite are not quite complementary, but it's one color outside the complementary, so to the left or to the right. That's called split complementary, so that's more comfortable for the eyes. And then it's best actually for good contrast is to use triadic, which is a triangle equidistant. So like the primary colors we know, red, blue, yellow, are going to be good colors to use. So here's what I mean by the complementary ones. So red and green, purple and yellow, orange and blue. It hurts the eyes because the contrast is too high, and especially for green, red, green, colorblind people, it's just going to look gray. Another thing to look out for are gradations. Be careful that the colors you use will translate in black and white printing. So there's grayscale, but there's other types of scale that are far apart. The colors can be seen. So the colors next to each other aren't very close. They're actually very different colors. So you can put them in consecutive bars and it won't hurt your eyes, and you'll see what the bars mean. So there is a website called colorbrewer2.org, which helps you find colorblind safe combinations of colors, especially for gradations. And something to be aware of is the color page cost. Sometimes journals charge you for using color, for printing in color, because they have to separate the color CMYK for the 4 different colors. So it's quite expensive. Now for the text, you have to sound professional. So when you describe your graph, don't just, the title, don't just say what the X and Y axes are. So it's better to actually have a descriptive title and say what the data are about. In the text, don't say see above, see below, because the page or the online page might be different. It might not be see above, see below. And don't say vague things like, oh, we compared the data statistically. So do summarize important data. For example, Group A achieved significantly higher mean SD scores than Group B, and then in brackets put the summary data, 75% versus 50%, and then the p-value, and then where the figure is. So avoid figure 2 shows this, figure 1 shows that. Give the actual message, and then table figure is at the end of the sentence. Here, of course, it's poor style. It's a very long sentence and some long words and the wrong formality. So keep it short and sweet and the right formality. So don't say things like skyrocketed or plummeted. Just say things simply and technically. The sample viscosity was initially high, but sharply decreased at 10 minutes. Similarly here, the style is wrong, but also the comparisons are wrong. So if you're saying thermal stability and you're comparing it in groups A and B, you're comparing thermal stability of A with, not directly with B, but the thermal stability of B. So you have to say the thermal stability of group A samples was significantly higher than that of B samples, and then you give some statistics. Note that we've used than, higher than. If you wanted to use compared to actually says the things are similar. So you want to say compared with to show that things are different. You can also say the thermal stability of samples was, and then that's the terms to the left. Then your comparison is actually comparing A and B. The thermal stability of samples was significantly higher in group A than in group B. So that's another way of doing it. Now, when you have a row of numbers and it's a list that belongs to another list, so A, B, and C, the order matters for these numbers, then you need to use the word comma respectively. And note you have to use similar, the same decimal points, the same decimal places, 1.0, 2.1, 3.0, and repeat the units to be safe. Or you can say the mean lengths were 1.0 centimeters in group A, 2.0 centimeters in group B. So it's like you're listing a table. You don't need respectively here, but be careful, decide whether you want to list it like this or whether a table is more appropriate. Or you can use the singular and say the mean length was 1.0 centimeters in group A, blah, blah, blah. So you can use plural or singular depending on the rest of the sentence. So finally, some do's and don'ts. So the don'ts are don't ignore ethics and don't commit FFP, fabrication, falsification, plagiarism. Be honest in your reporting, get permission and consent to collect, share, and publish data, and copyright permission if you have to reproduce a figure or reproduce a table from a published source. Don't manipulate figures, so no secret splicing of gel tracks, no cropping or local contrast changes to try to hide something in a photo. Don't stretch the axes of the graphs, and always start the axes at 0 at the origin, and be prepared to show all your original files to the journal editor. Don't manipulate your analyses. So usually though, you need to link where the full protocol is. So your protocol, data management plan, statistical analysis plan, all have to be planned in advance so that you don't do any fishing trips. You're not just testing lots of data to get significant p-values. And be careful of declarative titles in figures and tables. So sometimes people put conclusion titles as the title, but of course the conclusion might not be correct all the time. There's many caveats and conditions. So it's different from when you're doing a poster for a conference where you want to be quite aggressive and get people's attention. So watch out for journal style. Sometimes it's okay. Sometimes journals don't allow you to put declarative conclusion titles. Don't forget to check all the numbers add up. They add up to 100%. State the denominator n value. Check the precision, decimal places are consistent, and that you've stated the units. Don't repeat information in the text and tables or in different types of illustrations. And don't forget to check the overall message of a graph. If it shows an upward trend, it's easy sometimes because you revise your graph and then you end up saying downwards by accident, or something goes up, but the data actually shows it goes down. So that's a very common error. And finally, don't forget to check the illustrations in journal style. Many little things like formatting and alignment and how to label things, and even resolution. So for print, you need 300 dpi for high resolution photos and figures. Some do's. So do check your illustrations are easy to read, no 3D effects, clear labels, and follow the referencing order of the main text. Check the illustration numbering system that the journal uses and how they abbreviate figure to fig, whether they use a period, full stop after fig, how they number them. And also the figures and tables are independent. So if they come in a different order, you label and cite them in the order that you present them. So figure 1 might come first, and then table 1, then figure 2, then figure 3, then table 2. Check the illustrations suit the type of data. Remember, continuous, discrete, categorical. Get a professional to help you with artwork if you can. If you're lucky, the journal might help you redraw your figures and tables. And use colors that will translate in black and white printing. And check your illustrations as standalone. So the legends actually are very important because editors and reviewers sometimes do look at the figures and tables first before reading your text, because they can get the full message from the actual title and illustration and then the legend. And don't forget to define your abbreviations, or better still, avoid abbreviations. Describe the data source and the tests that you used for analysis. Don't assume normality. You might have to include a line saying that you tested for normality, and explain if there were multiple comparisons and that you corrected for those. And finally, present negative and unexpected data as well. Don't try to just delete outliers and hide them. So it's best to present everything and be transparent and complete, and submit your raw data as well after it has been anonymized, because many journals want you to share your data as well on a public repository. So we've had a quick tour through tables and figures and how to describe your data in the text. So now we've come to questions and answers, the Q&A. So please type your Q&A in the Q&A box, and I'll get to them now. So any questions you have about preparing tables or figures or graphs, or how to submit tables and figures to a journal? many people ask me, actually, how do you prepare the figures? So we looked at many examples. So we actually didn't prepare them, but we looked at ready-made ones. So it could be a completely separate webinar of many hours to show you how to use the software. So there is, of course, Excel is a very good program and does quite acceptable graphs and tables, but be careful of the either color combinations or the grid lines. So usually journals don't want complete grid lines that go across and down, or they want finer hairlines, or they want shading for every 5th row or 10th row or every other row. Actually, online journals don't seem to mind. They use those transparent cell lines to, so you do have a line separating every cell, but it's like a transparent version, or it's a very fine line. But there are some programs that your university might have. So I've listed some. So MATLAB, of course, is a very commonly used one, and GraphPad and GraphPad Prism. There's GraphViz. There's Matplotlib, which uses Python, or ggplot2, which uses R. And one that came up a lot when I did searches is Origin and Origin Pro and Kaleidograph. Now, statistical packages like Stata, SPSS, Minitab should also have a function to help you plot the data. There's also SigmaPlot, dplot, and Plotly. But some of those I think you do have to pay, or you have to pay for the premium version. But there are some free versions that I found, MetaChart or Graph or GNUplot, GNU, or LabPlot, or one called Orange. So check those out. There's one also Infogram, infogr.am, and one called Views, VEUSZ. So they take a while to learn, so do ask around in your group or your supervisor or other researchers, what do they use. Oh, I mentioned also before that don't assume your data are normal, so don't use t-tests, student t-tests for data that might not be normal. If it's skewed, if it doesn't follow a symmetrical bell curve, you can't use parametric tests, and you might have to show that your data did follow a normal distribution first. Something else you do have to show as well is the power calculation, sample size calculation, to show that the sample isn't too small and that your denominator n makes sense for your analyses. But two common tests for normality are the Shapiro-Wilk test and the Kolmogorov-Smirnov test. But if you go to Wikipedia, you'll see those two tests. So do tests for normality and present those data, sometimes in a footnote, to show that your analysis is the correct type and that you can present means and standard deviations. Otherwise, you have to go to medians and range or interquartile range. So how much do color pages charge? So different journals, if it's print, they might charge for color pages, or they might say you're only allowed one color figure, and any more than one or two you have to pay. So the charge will vary. Some journals I've worked for have been quite kind, and they say that if it doesn't matter, you can give a black and white version. We'll print a black and white version. But the online version, because it's online, it's free, you can put the color version online. So sometimes a photo or a graph, the color version would go online, but the print version would have black and white. But note that it's not really black and white. If you used a photocopier, you'll notice that the contrast isn't nice. it's very dark or very light. So it's not really black and white, it's more grayscale. So what they mean is use the grayscale conversion in Photoshop or whatever program you use, and then test it out to see whether that shows the features that you want to show. It might not work out. For example, if it was like a map with different colors, everything would just look gray. So sometimes you do have to use color charges and pay for that. Online journals, you wouldn't need to pay for color charges. Another thing about print journals, for whether it's color or not, you have to test that your figure shrinks okay, because print journals are confined to 1 column or 2 columns, so they will decide whether your figure will fit 1 column or 2 columns. So you have to do an experiment, use a photocopier or shrink down your figure and print it out yourself, and use the same measurements as the journal page to see whether you can still read your text. So the footnotes shouldn't be very small. And the font you should use, because it might look quite muddy and cramped, is to avoid the serif fonts, the fonts that have little feet on them. So Times New Roman has little feet. So use a sans serif or a non-serif font for figures and tables, because the lettering comes out much cleaner. So things like Arial, Helvetica, Cambria, Calibri would be good fonts to use in figures and tables. Okay, so we've come to the end of the webinar. And so I'd like to thank you for joining us, and a recording will be made available in due course. And you can find other free webinars and free educational resources at the AsiaEdit.com website. And I hope you found this webinar useful. Until next time, I wish you success, and goodbye, and stay safe.